Good morning, class. Today, we will be diving deep into the fascinating world of cyclic nucleotide metabolism. Within the human body, cyclic nucleotides, such as cyclic adenosine monophosphate, CAMP, and cyclic guanosine monophosphate, CGMP, play crucial roles in various cellular signaling pathways. Understanding the intricacies of cyclic nucleotide metabolism is essential for comprehending the underlying molecular mechanisms behind many physiological processes. To begin our journey, let's start with a brief overview of cyclic nucleotide metabolism. Cyclic nucleotides are small, intracellular signaling molecules that are synthesized upon activation of specific enzymes. They serve as important second messengers, transmitting signals from extracellular stimuli, such as hormones and neurotransmitters, to intracellular targets. The balance between cyclic nucleotide synthesis and degradation is tightly regulated, ensuring precise control over cellular responses. Now, let's focus on the process of CAMP production. Adenylate cyclus is the enzyme responsible for catalyzing the conversion of adenosine triphosphate, ATP, to CAMP. This enzyme is activated by various stimuli, including G-protein coupled receptors, GPCRs, which are integral membrane proteins that bind extracellular ligands such as hormones. Upon ligand binding, GPCRs undergo conformational changes that activate associated G proteins. These activated G proteins then stimulate adenylate cyclus, leading to increased CAMP synthesis. Moving on, let's explore the activation and function of the CAMP-dependent protein kinase A, pKa. pKa is a serine/threonine kinase that is activated upon binding of CAMP. The binding of CAMP causes the regulatory subunits of pKa to dissociate from the catalytic subunits, thereby enabling the catalytic subunits to phosphorylate specific protein targets. This phosphorylation can lead to changes in protein activity, gene expression, and cellular functions. Continuing our discussion, let's delve into the role of phosphodiesterases, PDEs, in cyclic nucleotide metabolism. PDEs are a diverse family of enzymes responsible for the hydrolysis of CAMP and CGMP to their respective 5-monophosphate forms. By degrading cyclic nucleotides, PDEs tightly regulate their concentrations, thus controlling cellular signaling pathways. Additionally, different isoforms of PDEs exhibit distinct tissue distribution and kinetic properties, providing further complexity to the regulation of cyclic nucleotide signaling. Now, it's time to shift our focus to CGMP signaling and guanylyl cyclus. Similar to CAMP, CGMP is an important second messenger involved in numerous physiological processes. Guanylyl cyclus is responsible for the synthesis of CGMP from guanosine triphosphate, GTP. Different isoforms of guanylyl cyclus exist, including soluble guanylyl cyclus, SGC, and membrane-bound guanylyl cycluses. Activation of guanylyl cyclus leads to increased CGMP levels and subsequent activation of downstream effectors. Let's now discuss cyclic nucleotide-gated ion channels. These ion channels are unique, as their opening and closing is regulated by the binding of cyclic nucleotides. When cyclic nucleotides bind to these channels, conformational changes occur, allowing the flow of specific ions across the cell membrane. Cyclic nucleotide-gated ion channels are found in various cell types, including photoreceptor cells in the retina and olfactory sensory neurons, where they play crucial roles in sensory signal transduction. Next, we will explore the intricate crosstalk between CAMP and CGMP signaling pathways. While CAMP and CGMP primarily regulate different targets and cellular functions, there are instances where these two second messengers interact. For example, CGMP can modulate CAMP signaling through phosphodiesterase-regulated pathways. Furthermore, crosstalk between CAMP and CGMP has important implications in cellular processes such as cardiac function and smooth muscle regulation. Moving forward, let's study cyclic nucleotide phosphodiesterase inhibitors. These compounds are pharmacological agents that block the activity of specific parade isoforms. By inhibiting PDEs, cyclic nucleotide levels can be regulated, leading to altered cellular signaling pathways. Phosphodiesterase inhibitors have therapeutic potential in various diseases, including cardiovascular and respiratory disorders, and are commonly used in the treatment of erectile dysfunction. Now, let's focus on the role of cyclic nucleotide-dependent protein kinases as substrates for phosphodiesterases. pKa and protein kinase G, PKG, are two major cyclic nucleotide-dependent protein kinases that are substrates for PDEs. Phosphorylation of pKa or PKG by PDEs can modulate their activities or stability, thereby impacting downstream signaling events. Understanding the interplay between cyclic nucleotide-dependent protein kinases and phosphodiesterases is crucial for deciphering the complexities of cyclic nucleotide metabolism. Shifting gears, let's examine the role of cyclic nucleotides in cardiac function. Both CAMP and CGMP play critical roles in the regulation of cardiac contractility, relaxation, and overall cardiovascular function. Dysregulation of cyclic nucleotide signaling pathways can contribute to the development of various cardiovascular diseases, including heart failure and arrhythmias. Pharmacological interventions targeted at modulating cyclic nucleotide levels show promise in the treatment of cardiac disorders. Now, let's turn our attention to the cyclic nucleotide-mediated regulation of smooth muscle. Cyclic nucleotides play significant roles in maintaining the tone and contractile properties of smooth muscle, which is essential for various physiological processes, including vascular homeostasis and gastrointestinal motility. Activation of cyclic nucleotide-dependent protein kinases and the downstream effectors they regulate are major contributors to the modulation of smooth muscle contraction and relaxation. As we near the end of our journey through cyclic nucleotide metabolism, it is important to highlight the genetic and epigenetic regulation of this intricate process. Various genes, including those encoding adenylate cyclus, guanylyl cyclus, and parade isoforms, exhibit tissue-specific expression patterns. Additionally, epigenetic modifications, such as DNA methylation and histone modifications, can influence the expression and activity of genes involved in cyclic nucleotide metabolism, adding another layer of complexity to this field of study. In conclusion, the study of cyclic nucleotide metabolism is crucial for understanding the molecular mechanisms that underlie many physiological processes. From the synthesis of cyclic nucleotides to their degradation and the intricate signaling pathways they modulate, each aspect contributes to the precise regulation of cellular responses. As we continue to unravel the complexities of cyclic nucleotide metabolism, new insights into their therapeutic potential and implications in various diseases will emerge, enriching our understanding of human biology.